Welcome. My name is Jacob Best. I'm a second year biology student here at the University of Florida, and today we're going to talk about worms, but not the kind you fish with. No, today we'll be talking about the nematode, Caenorhabditis elegans, or C. elegans for short. C. elegans makes a great model organism because we've mapped all of its over 20,000 genes. Hence, it has been used to study many fundamental biological processes, including cell death and development, microRNA and siRNA biology, neurobiology, and today's topic, aging. Aging is a fundamental biological process that affects all eukaryotes to one degree or another, and C. elegans is no exception. Indeed, because of its short lifespan of about 3 weeks, it makes for a great organism to study. So, to assess the role of each gene in the aging process, we prepared an interference RNA, or RNAi, plasmid for each and every gene in C. elegans. In this way, every gene can be silenced individually. Next, we allowed E. coli, C. elegans' food source, to store these plasmids for later feeding. Meanwhile, we collected worm eggs and raised them to hatchlings. We then plated these hatchlings onto the E. coli and allowed them to grow to adults. These adults were then given an FUDR treatment to prevent them from laying eggs, and were allowed to grow for an additional 2 weeks. At the end of the 2 weeks, we assayed the worms with blue dye, hence the Smurf moniker, and used intestinal leakage, or permeability, as a marker of aging. Worms with dye leaking through their whole bodies are said to be short-lived mutants, and those with little leakage are said to be long-lived mutants. As an example of how this works, let's consider the mutants shown in Figure 1. The left column shows what dyed worms look like after 1 day of eating the RNAi, and the right shows what they look like after eating the RNAi for 15 days. DAF2 is considered a long-lived mutant. As you can see, there's not much leakage on day 15. Next, we see the control, which is shown some leakage, but not a lot. Finally, we look to HSF1, which is almost completely blue. The more blue a given mutant has, the more confident we can be that the gene we knocked out was crucial for a long and healthy life. And the converse can be said of long-lived mutants. Assessing an entire genome can take time, so more screening is still necessary. But we expect that genes that contribute most significantly to the modulation of intestinal permeability affect aging, which is intriguing in and of itself. But perhaps more interesting is that about 80% of C. elegans genes have human lookalikes, or homologs. And on top of that, most, if not all, aging related genes are evolutionarily conserved. So learning about these worms, it's like learning about ourselves. Thank you.